Good evening, everybody. We are group number 1, and our project is solubility prediction. So, in this reading group activity, we were supposed to read 3 research papers. Out of that, first one is the aqueous solubility prediction of drugs based on molecular topology and neural network modeling, done by J. Hughes Conan, then M. Salo, and J. Tuskinen. So in this research paper, the question about the research paper is, can aqueous solubility of complex drug-like compounds be accurately predicted using artificial neural networks? So first of all, let's see what is the importance of aqueous solubility in drug production. So the drugs should be aqueously soluble to absorb as well as biologically available. To determine the aqueous solubility of the drugs, traditionally used 2 methods. They are laboratory testing and then the theoretical predictions. There are problems with those predictive method. In laboratory testing, it takes long time as well as huge cost. On the other hand, the theoretical method, it is applicable only for small molecules. So in that context, the pharmaceutical industry has entered to computational methods to find the solubility in drugs. In this research paper, the application of artificial neural network is discussed. So, when we consider about the methodology of this artificial neural network, the data set used for this ANN is includes topological indices calculated using Malcolm C software. Out of those data set, 75 are connectivity and shape indices, and then 26 are atom type state indices. From those 101 molecular descriptors, 23 were selected via sensitivity analysis. So here is the details about the neural network, and it is a 3-layer input, hidden, and output neural network. Then the standard backpropagation is the training algorithm used, and here are the parameters of the neural network. So software called NeuDesk is used here to training the neural network. Then here is the training strategy is the training until the mean squared error reached to an optimal threshold. So here are the results of the neural network. So when we consider about the findings of the neural network, it is when we consider about the findings of the research paper, this newly made artificial neural network is compared with the existing models such as Klopman's group contribution model. However, this ANN is underperformed in compounds outside the training domain. So when we consider the domain-specific performance, this ANN model work well for compounds structurally similar to the training set. However, the prediction quality dropped for molecules with unusual groups or very low solubility. At the end of the research paper, we can come to a conclusion that a practical and reasonably accurate model for predicting aqueous solubility of diverse drugs-like compounds can be built using ANNs. However, there are limitations such as reduced performance outside the structural domain, as well as lacks 3D structural awareness. Then it also fails on compounds with extremely solubility behavior. So now I invite.
Well, I have a question for you. Is your model interpretable? So, the next model we are going to talk about is actually interpretable model. Interpretable model. So here we are going to discuss about what actually affect the solubility of a molecule. So here the main research gap we have identified was the ML models such as ANN. It only tries to predict the solubility, not try to identify why it is soluble or why it is not. So the main thing is topological shape, like 3D structure of the model is not considered in those kind of ML models, and also descriptors like molecular weight and the 3D structure are both not used in, actually they were separately used, but they were not used together. So then our methodology is we have used Wang dataset, it's a publicly available dataset, and then here actually we have used topological data analysis in two methods, map algorithm and persistent homology. In map algorithm, we are trying to map our high dimensional data into overlapping sorry. Into overlapping clusters. So then we are clustering our data into, here we are replacing our data as clusters, and the overlapping shared data points are represented as edges. So now we can see the structure of the data as a visual structure. So in the persistent homology, we are trying to zoom out from, in a 3D structure, we are trying to zoom out from, starting from atom, we are zooming out. So we are looking like the rings in the molecules, and then we are zooming out and test how the solubility affects. And then we have found that the number of rings, actually this is a simple ring we have seen in molecules, and these rings actually affect the solubility, and also the halogens, the seventh column of the specifically the chlorine, the molecules with two rings and chlorine atoms are much less soluble. Also, then the molecular weights actually affect the solubility as in simpler molecules, but as the molecule grows, the effect disappears. And the important thing is persistent homology and the mapper algorithm actually agrees in those key features. And then the conclusion we come to that, the TDA reveals the meaningful patterns beyond traditional ML. Actually, it says why these molecules are soluble or not. And then the shape and structure, those are just not values. Actually, it matters in solubility. And then we have topological methods explaining why, not just predicting why the solubility is happening. Then this approach helps in early stage drug decision and screening. Now I would like to invite Kaushita.
So, if you look at the new paper, it's so demotivating after they were so optimistic about their models. So, let's look at this paper now. Will we ever be able to accurately predict solubility? So, why the state-of-the-art models that they discussed fail in real-world scenario? So, Satha, did you test your model on an external data set? On your own data set. Did you test your model validate your model on an external data set? So that's the main issue. These models, they were not validated in a reliable external validation test set, and their metrics are over-optimistic. They record the best case scenario in their own data set, the validation set that they set aside. And the other research gap, the applicability domain. So in the chemical space, the region where your model performs the best, the most confident region, is known as the applicability domain. None of these models mentioned about the applicability domain, or when they mention it, it's mostly in an improper way. And the next gap, the lack of analysis on the data quality before training the data set. So you take the training data set and start training without analyzing the data quality of each data set. So what I found, the state-of-the-art models perform poorly in the real-world scenarios due to poor data quality. Not the model, but the poor data quality. So specifically, public data sets contain problematic data. So what is this problematic data? When we talk about thermodynamic solubility, it has intrinsic, apparent, and water solubility, 3 types. But these 3 types are mixed in public data sets. And none of these models tell us which kind of thermodynamic solubility that they are concerned of. And we have duplicates in these data sets because when these data sets are supersets of smaller training data sets, and one training data set might contain the same sample that's existing in another. So now there's data leakage happening, and in our own validation set, we also have a training sample. So now we are over optimistic about our data. We create our own data and then we validate on a similar sample that's in the training set. Then the measurement noise. So the measurement noise strongly correlates with the model error. We get this training data from the measurements. And we have the interlab standard deviation. Higher standard deviation indicates that this experiment is poorly reproducible. And that creates a lower bound on our accuracy. So no matter what our model produces, model predicts, our accuracy is bound by this standard deviation in interlab experiments. Next, to mitigate this, we curated a data set. So this significantly improved the model performance on internal validation sets. So all good then? No. So accuracy even varies within the compound type, the ionization state. The deuteriums had the lowest prediction error, so the highest accuracy there. And uncharged compounds had the highest prediction error. And we see intermediate error in singly charged compounds. And most importantly, even with the curated data set, this external validation reveals that these models perform really bad in generalization. They do not generalize very well. So the next thing is the applicability domain, the region in the chemical space where our model is confident. We can reduce the error by converging into a small area. However, in that case, our model is not applicable in the real world scenario because our range is so small, but the error goes down. So it's a trade-off. So that's the definition of applicability domain. And finally, we found that no matter the ML model, there had been different neural networks, transform networks, autoencoders, no matter which sophisticated architecture, no model consistently perform well. It's similar to Nolan's theorem, no freelance. So then we produce a decision tree workflow for any data set curation that can improve the internal validation performance. But remember, not the external validation. So this is our workflow. So you validate the remove samples based on the structure. If it's a valid structure, you keep it. Then the condition, you remove all the buffers, co-solvents, excipients, and keep the experiment range in the pH 7 to 6 to 8 range. Then the measurement, the experimental measurement should have a standard deviation less than 0.5 log units. Then the composition should be monoconstituent, not mixed compositions. Then the technique should be, this is a standard technique. If the model has higher solubility, then we have to do the shake flask method. If less, it's a slow stir or column elevation method. So in conclusion, yeah, we are pessimistic, but this work produces a clear direction. So robust data curation, external validation, and AD, applicability domain aware modeling, is essential if we are to use solubility prediction in the real world scenario. And in concern of our project, so we first found out that nonlinear models, specifically the ANN model, perform better than the linear models. And then we found out that focusing on the interpretability of the model also helps on the performance, the accuracy of the prediction. And then finally we found out that these SOTA models are not generalizable, and we have to look at the data again, robust data curation, external validation, and AD-aware modeling. So that's it for our presentation. Any questions from anyone? Hopefully not. Ask easy question, you can get 5 marks. You can. If you want, we can change the slides.
Questions? We'll give 2 minutes to ask questions.
So if you can go to the first paper, you showed some parameters you chose for the model there. So how did you figure out that these are the parameters? So how did you come up with these parameters?
These parameters were stated in the research paper.
Yeah.
So we wrote the paper. So it means that first we implemented the neural network with the data set given in the research data set found. Then these parameters were tuned, and then finally get the appropriate ANN. So that's.
So you're talking about like these models will predict do good on data. So we'll fail on these other external data. So what is the like the core difference between this external data and the data here? Like, do you have enough external data so that you can use it, those data to train?
So the whole idea of machine learning is to learn a generalizable model. So it should perform on both the training data set, the training in-domain data sets, and also the out-of-domain, out-of-distribution data sets. So for training, if you are taking one training data set, even let's say the same similar compounds, it should also perform the similar metrics in a independently found, independently experimented another data set. So that's the whole idea of generalization. Is that clear?
You are saying that the domain is so vast so that it's
Yeah, so that's where the applicability domain comes in. You have to specify which area in the chemical space that we are concerned with. So if we cover space in the chemical, so they have found out methods to draw out the chemical space. That I didn't explain that. So in that chemical space, if our model is trained on a certain region, so then our model should be generalizable in that AD to any data set that's within that same chemical space. So that's the whole idea of the applicable domain. So it's, as you said, it's not possible to train on a small set and then generalize to entire chemical space. So I don't think that's possible with machine learning. And so that's why we restrict into small applicability domains. Hard word to pronounce.
In that sense, what is the purpose of this dataset curation?
Yeah, so the dataset curation happens. So the dataset curation is based on these two problematic data scenarios, heterogeneous data and duplicates. So in datasets we have heterogeneous, like different kinds of solubility, intrinsic, apparent and water. We remove the intrinsic and apparent samples and keep only the water. And then we have these datasets are supersets of smaller training datasets from different labs. So each lab has conducted the same experiment on a compound, and there's another lab done another experiment on the same compound. And when we create the superset, we don't know that these are the same compound. When we take out the validation set also has the same compound. So we train on the same compound and again validate on the same compound, and we like put out our performance as very high. But in reality, that's not the scenario. We have to get rid of all those duplicates and heterogeneous samples and train on an independent training set, then validate on an external data set. So in fact, these when the curated with data curation, it significantly improved the model performance on cross-validation within that data set. So about 0.1 log units. I hope.
So what's the difference between internal and external?
Yeah, so I think you didn't understand really. So these labs, let's say we have a lab here, we have a lab in University of Colombo and University of Peru. So we train independently on our data on similar compounds in our data in our lab within that applicability domain. So then University of Colombo also had done the similar experiments, different compounds, but within that applicability domain. So then we train on our data set in Peradeniya and test on the data set in University of Colombo. But what happens with these guys' cases, we perform the experiments in University of Peradeniya, keep out the set for validation, train our model with our data, and then test on the same data set that's curated from University of Peradeniya.
On the second paper, do they mention anything about the computing requirement? Because it has good number of features and
Yeah, actually, here the feature numbers are 1000, this type of features. So in map algorithm, they have mapped using selected some features and then map like that. They have mapped several times. Then in persistent homology, actually the data set is actually the data features is not considered. Actually, the structure of the molecules are considered. So here we are actually considering both the features we have and the 3D structure separately, and then after analyzing we found that these two cases give the same answers.
so, do they mention anything about the computing power requirement for this experiment?
Yeah, they have mentioned about softwares they have used and the data processing part.
So where the interpretability is coming from in this case?
Interpretability is coming from actually they are trying to say like not just focusing on accuracy of like to they are like trying to
How are they making them interpretable?
so here the data set actually that's why I mentioned we are trying to get the features, both features and the 3D structure. Previously we were using only features, then using features we are predicting. Now we are adding also the 3D structure too. Then with the both methods giving the same results. They have found the same result like they have specifically mentioned that the molecules with 2 rings and chlorine atoms, they have specifically mentioned that both the 3D structure, persistent homology and map algorithm, they have given the same result considering that 2 rings and chlorine atoms. Is it good answer?
So the interpretability is coming from looking at the correlation of
Yeah.